So, welcome to everyone. A very good morning, good afternoon, good evening, and good night, depending on the time when you are watching this video. So, in this video, we will going to talk about a special characteristics of the genome of the influenza virus that create a lot of problem to us, especially to the vaccine developers. So, this special type of features is known as genomic plasticity. Okay, and this special characteristics ultimately leads to antigenic shift and antigenic drift. That is our today's topic of discussion. Okay, so genome plasticity means the genome of influenza virus changes frequently due to point mutation and reassortment of the RNA segment. Okay, because we have told you that influenza virus consisting of 8 negative sense single stranded RNA virus, right? So as they are segmented, there can be chance of reassortments, okay, that ultimately causes change in the genome of the RNA virus. Okay, how it is going on and what kind of complication it creates, we will going to discuss. So let's move. So genome plasticity is one of the most important feature of the influenza virus, that it leads to both subtle, that means small, and sometimes drastic changes in the viral protein, and that is obvious, right? Because if genome content changes, that may be due to either point mutation or reassortment of the genome, it ultimately reflects a change in the viral protein. Okay, and due to this change, there can be a alteration in the virus antigenicity. Okay, so if this change is smaller, then antigenic drift occurs, and if the variation in the protein is significant, then antigenic shift occurs. Okay, so our first topic will be antigenic drift. Okay, so remember, antigenic drift occur due to some type of mutation. That may be point mutation, that may be anything. Okay, so antigenic drift, this is the definition of the antigenic drift. Antigenic drift is the result from the accumulation of mutation of HA and NA in a single strain of flu virus within a geographic region. This usually occur every 2 to 3 years of the causing the local increase in the number of the flu infection. Okay, that means antigenic drift causes mutation in the genomic content of the influenza virus that ultimately facilitates mutation in either hemagglutinin or both hemagglutinin and neuraminidase of the single strain. Okay, so it is a slight change in the genomic content of the influenza virus, and this do not leads to formation of any kind of new strain. Okay, that means this is the small variation in proteins that we have talked about. Okay, this usually occur every 2 to 3 years, increasing the number of influenza infection in local. Okay, so remember it, that antigenic drift is the accumulation of the mutation that changes hemagglutinin or neuraminidase or both hemagglutinin and neuraminidase. Okay, so let's consider this picture. This is a influenza virus. Okay, this influenza virus have the ability, this influenza virus is going to infect the person A, and during the interaction with the receptor that is present on the surface of the person A, which is terminated with sialic acid, we will going to talk about what kind of receptor influenza virus prefers for their interaction and then entry in when we will talk about we will going to talk about the replication and life cycle of the influenza virus. So for now, let's remember that this is the receptor which is specific for the influenza virus. Okay, so and remember, hemagglutinin initially play the role for the entry of the influenza virus. Okay, so now when this influenza virus try to enter in the cell of the person A, during that time the antibodies which is secreted by the immune system or which is produced by the immune system of the person A inhibits their entry, okay, by preventing the interaction with the hemagglutinin and the receptors, okay, and as a result this influenza virus cannot infect person A. Okay, now let's consider another situation where the same influenza virus that I have explained you here infects person B. Okay, and we all know that after infection, after entry, they will release their genomic material in the cytoplasm. Okay, and this genomic material will go to the nucleus and replicates with the help of the host cell machinery, right? So during this replication, let's say a genomic mutation occurs that may be due to anything. For example, let's consider a one example which is common example, that is erroneous insertion of the nucleotides due to RNA polymerase or by RNA polymerase. Okay, and as a result this yellow color portion or yellow color nucleotide is incorporated. Okay, so why this mutation is occurring? This mutation is occurring due to, let's consider, erroneous insertion of the nucleotides by the RNA polymerase. Okay, now after the replication there will be packaging where those genomic material will be packaged inside the capsid. Okay, so these wrong nucleotide containing genomes are also packaged inside the capsid. Okay, as a result what happens, this ultimately leads to a modified virus. Okay, and due to the presence of that new nucleotide, it ultimately reflects in its protein content. Okay, and as a result, let's say this gene is responsible for the synthesis of hemagglutinin, so as a result the hemagglutinin also changes. Okay, that means during the replication step, due to the erroneous base insertion or erroneous insertion of the nucleotides by the RNA polymerase, a mutation has occurred. Okay, now when these genomes are incorporated inside the capsid, this gene will produce protein. Okay, and here we are considering that the protein is hemagglutinin. Okay, and as a result this hemagglutinin acquires mutation. Okay, clear? Now under such circumstances, remember it, these do not lead to the formation of a new strain. This is just a slight modification in the hemagglutinin, and that's all. Okay, that is the difference between antigenic drift and antigenic shift. Antigenic drift do not leads to the formation of new strain, but antigenic shift facilitates the formation of new strain. We will going to talk about. So now under such circumstances, when this new viral strain, when this modified viral strain, I should say, going to infect the person again. So here I have forgotten to mention here the yellow color, so here should be a yellow color. Okay, so What do you think? As now the hemagglutinin is modified, can the antibiotics, sorry, not antibiotics, it is antibodies, can the antibodies can recognize the hemagglutinin anymore? Because the antigen binding portion of the antibodies, known the red marked portion of the hemagglutinin, right? But here the red marked portion is totally absent, because here instead of red marks there are yellow portions. Okay, so as a result, the antibodies from the cell, or antibodies which is coming from the host immune system, cannot protect the host from the infection of the modified viral strain, right? Because antibodies cannot help us here again, because as the hemagglutinin is slightly modified, they are unable to recognize. Okay, so that is the idea of antigenic drift. Antigenic drift is nothing but the accumulation of the slight mutation of either in HA or NA, or in both HA and NA. That may be due to some kind of mutations. Okay, we have talked about one type of example, that is insertion of the erroneous base by RNA polymerase. Okay, that ultimately causes a change in the hemagglutinin, and as a result, the antibodies that the host immune system develops, especially for that hemagglutinin, cannot recognize the hemagglutinin anymore. Okay, so remember it, in antigenic drift small mutation occur but not causes change in the strain. Okay, that means the strain is exactly the same, this is a small mutation. Okay, so if we consider an example, so here such kind of things occur, that due to erroneous addition of nucleotide by RNA polymerase, there is changes in neuraminidase and hemagglutinin. So that is the concept of antigenic drift that we have talked about. Okay, so it is occurring due to genomic plasticity. Okay, this is occurring due to change in the genomic material that may be due to some mutation. Okay, this mutation causes small changes in the viral protein, that may be in hemagglutinin or neuraminidase, or both hemagglutinin and neuraminidase, and as a result the virus can infect the host cell which it previously unable to infect due to the modified hemagglutinin. That is the concept of antigenic drift. Okay, so this is the GIF for this is the GIF for antigenic drift, where you can see changes in the surface protein of the virus, that may be hemagglutinin or neuraminidase, and as a result the antibodies of host cell cannot recognize them. Now there should be some drastic changes we have discussed. These drastic changes is due to the antigenic shift. What is antigenic shift? Antigenic shift is the large antigenic change that occurs due to the accumulation of the reassortment of the genome. Why reassortments occur? Reassortments occur when two different strains of influenza virus infect a same host cell. Okay, so as a result, let's say this is a example of host, and inside the host both the genomic content 1, 2, 3, 4, 5, 6, 7, 8. These 8 influenza viral genome from, let's say, virus A, and 1, 2, 3, 4, 5, 6, 7, 8. This 8 influenza viral genome from virus B is present in the same host cell, and now during the packaging, there is a reassortments occur. That means mixing occurs, and as a result, let's say among 8 RNA genome, 4 RNA genome which is red marked is transferred to the green marked, and 4 RNA genome from 4 green RNA genome is transferring to the red marked. As a result, a new virus called virus C is formed, having 4 red RNA genome and 4 green RNA genome. Okay, and as a result, it ultimately facilitates the accumulation or formation of new viral strain. Okay, and that obviously causes changes in the hemagglutinin and neuraminidase. Okay, but here the change is drastic, because remember, the 50% of the genomic content here is changed, right? Previously it was 8 red RNA segments, but now here 4 red RNA segments and 4 blue RNA segments. Okay, so that is the thing that is going on in antigenic shift. So remember, antigenic shift occur when two different viral strain, virus A and virus B, infect the same host cell, and inside the host cell during the packaging there is a reassortment of the RNA segment occurs. Reassortment means simply the mixing of the RNA segments. Okay, and as a result, it ultimately facilitates the formation of new RNA strain. So here it does not mean that there will be equal sharing, that 4 red RNA genome and 4 blue RNA genome will occur. There can be odd separings, mixing can also occur. That means there can be 5 red RNA genome, 3 blue RNA genome, or let's say 3 red RNA genome, 5 blue RNA genome. That kind of possibilities can also occur, which ultimately leads to the formation of new strain. Okay, that means simply the mixing of the genome from the virus B and virus A occurring when they are infecting the same host cell, and that causes a type of change which we are calling as antigenic shift. So antigenic shift is a large antigenic change resulting from the reassortment of the genome when two different strain of virus infect the same host cell and incorporated into a single new capsid. So remember, antigenic shift induces a greater change than antigenic drift. Antigenic shift is less frequent than antigenic drift. Okay, because here drastic change occurs, right? On the other hand, antigenic drift is just a slight mutation. Okay, so this is an example of antigenic shift that I have downloaded from internet from makegif.com. So there another strain called blood influenza A strain is coming inside a host, and there already a pig strain viral RNA is present. Okay, now there should be a mixing. Okay, this mixing ultimately leads to the formation of a new viral strain. Let's say this example, in this example, the pig is infected with a influenza virus from the pig, influenza virus from the bird, and influenza virus from the humans. Okay, now due to the genetic reassortments, there is a formation of new strain that contains proteins from pig, proteins from birds, proteins from humans, as you can see. So this is the concept of antigenic shift. So here we have talked about the genomic plasticities. This genomic plasticity is nothing but change in their genome that may be due to some mutation or maybe due to the genetic reassortments. When the change is due to mutation, let's say for point mutation, the changes will be a small change or slight change that ultimately induces a small change in their viral protein and ultimately the viral antigenicity. So this kind of situation is known as antigenic drift. On the other hand, the genetic reassortment occur when two different viral strain infect the same host cell and inside the host cell when there is a genome of two different viral strain and mixing of them occurs. Okay, this leads to the greater change or drastic change in the genomic content of the influenza virus that ultimately leads to the formation of new viral strain. Okay, and as a result it will create a drastic change in the protein content of the virus that ultimately causes antigenic shift. So that is the concept of antigenic shift and antigenic drift. Remember, antigenic variation occur almost yearly with influenza A virus, but it is less frequent in influenza B virus, and it is not demonstrated in the C virus. So that is why we are requiring influenza immunization yearly, because we have discussed, right, that may be whether the cause is antigenic shift or antigenic drift, there will be a change in the hemagglutinin or neuraminidase, right? And as a result, let's say we have made a vaccine for against a particular type of strain, but with time they undergo antigenic variation, and as a result the protein which is present on the surface changes, and as a result now that vaccine who have prepared neutralizing antibodies inside our body are of no use, because those antibodies cannot recognize the proteins which are present on the surface of the virus no longer. Okay, and as a result now we have to develop the vaccine again. That is the reason why developing of influenza flu vaccine is very difficult. The reason is very simple. The reason is genomic plasticity, which causes frequent change in the genomic content of the influenza virus. That may be small, that ultimately facilitates antigenic drift, that may be drastic, that ultimately facilitates antigenic shift. Okay, in both the cases the surface protein can changes. Okay, antigenic drift do not causes the formation of new strain, but antigenic shift facilitates the formation of new strain. And another important information, that antigenic shift facilitates pandemic. Okay, but antigenic drift do not facilitate pandemic, but they causes epidemic in between pandemics. Okay, so I hope this video is very clear to you now. So thank you for listening to this class.